Hi everyone, I'm Dr. Anjali Devi, Associate Professor, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research. I'm handling the course Enzyme Technology. Today, in this session, we are going to learn about comparison of chemical as well as enzyme catalysis as well as models of enzyme substrate complex formation. Coming to the first topic, comparison of chemical and enzyme catalysis. First, until now, we have learned about what is an enzyme. Enzyme is a biocatalyst. It's proteinaceous in nature. And what is a chemical catalyst? It is an inorganic molecule. So, nature-wise, a chemical catalyst is a simple inorganic molecule, whereas enzyme is a complex protein. Coming to the function, chemical catalysts are the substances that increase or decrease the chemical reaction but remains unchanged. Coming to the enzymes, enzymes are proteins that increases the rate of chemical reactions, converting the substrate into product. The functioning of catalyst is not controlled by any regulator molecule, but the function of enzyme can be regulated. Enzyme activity can be regulated by specific substances which can change the conformation of enzyme molecules. Catalysts or chemical catalysts can catalyze diverse reactions, whereas enzymes are very specific and they will be participating in the reactions with single or more than one substrates. Coming to the molecular weight, chemical catalysts are low molecular weight compounds, whereas enzymatic catalysts or enzymes are high molecular weight globular proteins with three-dimensional structure. Coming to the reaction rates, the chemical catalyst will catalyze the reaction very slowly, but these enzymes will catalyze the reactions very fastly. Coming to the specificity, the chemical reactions are less specific. They are not specific. That's why they will be producing a number of byproducts. But enzymes are highly specific. That's why they are producing specific products. And what are the conditions for chemical catalyst to work? This chemical catalyst works under high temperature, high pH and pressure. And they are not much influenced by short wave radiations, whereas enzymes operate under mild conditions like very low pH and less temperature. And these are denatured in presence of rays of shorter wavelength. And coming to the type of chemical catalyst, there are two types of chemical catalyst. One is positive as well as another is negative catalyst. And coming to the enzyme, there are two types of enzymes. One is activation enzyme and another one is inhibitory enzymes. What are the alternative names for this chemical catalyst? They are called as inorganic catalysts, whereas enzymes, these are organic catalysts. They are most probably called as biocatalysts. Both of them, chemical catalysts as well as the enzymes, they are involved in lowering the activation energy. In order to understand more about the differences between the chemical as well as the enzyme catalyst, go through this video.
Catalysis is the acceleration of the rate of a chemical reaction due to the participation of an additional substance, which remains unchanged, called a catalyst. It may be inorganic catalyst or organic enzyme. Wilhelm Ostwald's work with acidic and basic catalytic reactions led to him receiving the Nobel Prize in Chemistry. The term catalyst is derived from the Greek word kappa alpha tau alpha lambda epsilon epsilon iota, which means to annul, or to untie, or to pick up. Any reaction driven by the use of catalysts is called catalysis, a term coined by the Swedish chemist, Jöns Jacob Berzelius, in 1835. Jöns Jacob Berzelius' The discovery of catalysts allowed scientists to examine and understand the nature of various chemical reactions. The first instance of such an element being present in biological reactions was described by Louis Pasteur during his experiments with wine production. This biological element was later identified and termed as enzyme by the German physiologist Wilhelm Kuhn in 1878. The term is derived from the Greek word enzymos, which means in leaven. Since enzyme is a subset of catalysts, they both share various similarities. They are needed in trace amounts in a reaction. While they can accelerate the rate of a reaction by reducing the activation energy, they themselves cannot initiate a reaction. During the course of the respective reactions, both combine with the reactant or substrate to form a temporary complex. Despite this, the inherent composition and the quantity of these molecules remains unchanged, and hence can be used again and again. Also, the reactions that they catalyze are reversible in nature. Despite all these similarities, they do show distinct differences as well. Catalysts versus enzymes. Nature catalysts simple inorganic molecules. Enzymes complex proteins molecular weight catalysts slow. Enzymes high reaction type catalysts chemical. Enzymes biological specificity catalysts wide range. Enzymes highly specific efficiency catalysts less efficient. Enzymes more efficient regulation of function catalysts cannot be regulated. Enzymes can be regulated types catalysts positive catalyst negative catalyst. Enzymes activation enzyme inhibitory enzyme conditions required catalysts high temperature and pressure. Enzymes mild conditions of physiological pH and temperature example catalysts platinum, copper. Enzymes amylase, lipase catalysts and enzymes have a wide range of applications, not only in the natural environment and laboratory settings, but also in industries. Catalysts are utilized in energy processing and in the production of chemicals. Enzymes are used in food processing, breweries, wine production, biofuel industry, antibiotic production, etc.
So, until now, we have learned about what is an enzyme, what is the differences between a chemical catalyst and enzyme. Now we are moving towards to learn what is a substrate and what is enzyme substrate complex. So, what is a substrate? A substrate is a molecule upon which enzyme acts. So, just consider this as an enzyme and this as a substrate. Once the substrate is coming to the and binding to the enzyme, it is exactly binding to the active site of enzyme. And once it is binding, the substrate is binding to the active site of an enzyme, it is forming an enzyme substrate complex. So Arrhenius in 1888 explained the catalytic action of enzyme and proposed the formation of this enzyme substrate complex. And once this enzyme substrate complex is formed, a number of bonds in the reactants will be broken down, a number of new bonds in the products will be formed, ultimately leading towards the release of this product, and your enzyme will be released back. Now this enzyme will be changing to its original orientation, regaining its original conformation, and now your enzyme is ready to bind with another substrate molecule. Just see this figure. Just you can observe an enzyme and a specific site on enzyme called as active site to which this substrate is coming and binding. Now we are going to learn about various models of enzyme substrate complex formation. Now we have a question. How does the enzymes know whether or not the molecules next to them is a substrate that they want? There are two models that attempt to explain this question. The first model is your lock and key model that is proposed by Fisher in 1894, and the second model is induced fit model proposed by Koshland in 1958. So first we are learning about lock and key model. This model explains the high specificity of enzyme, but it does not explain the stability of the transition state that the enzyme achieves. Now you have to learn what is the transition state. When an enzyme and substrate is combining, they will be forming an enzyme substrate complex. This complex, the state in which this complex enzyme substrate complex is present, this is called as a transition state. Now we'll learn more about this lock and key model. According to this Emil Fisher, he have assumed enzyme and substrate as lock and key. For example, if you consider a lock, every lock will be having specific key, and the key will be having exact size and shape that fits into the lock. So he have assumed enzyme as lock and substrate as key. So this model suggests that enzyme and substrate possesses specific complementarity geometric shapes that fit exactly into one another. That means enzyme and substrate, they will be having complementarity and they fit with one another. Like a key into a lock, like a key into a lock, only the correct size and shape of the substrate would fit into the active site of the enzyme. Just see this figure. So in this figure, it's very clearly given that this substrate, the key, will be coming and exactly binding to the lock. There's nothing but substrate is coming and binding to the active site of enzyme. Just see this video once. You'll be understanding more about this model.
Lock and key theory of enzyme action. Here, the larger enzyme molecule comes in contact with smaller substrate molecules to form an intermediate complex. This intermediate complex or enzyme-substrate complex soon breaks to release the enzyme and product of biochemical reaction. Mode of enzyme action. Biosynthetic reaction. Here, the active sites establish contact with substrate molecules to form products of biochemical reaction. Mode of enzyme action. The enzymes bear distinct cavities specially designed for only specific substrate molecules with proper geometric shapes that can fit correctly.
We are moving towards induced fit theory. Now, what is this induced fit theory? This induced fit theory or induced fit model is nothing but is a modification of lock and key model and is generally thought to be more accurate version of lock and key model. The active site is not an exact fit for this substrate. Rather, the substrate induces change in the enzyme, causing it to modify its shapes until the binding is complete. That means, according to lock and key model, Fisher had assumed enzyme as a lock, and he said that the substrate will be like a key, which are very like your enzyme is very rigid, and it will be exactly binding to the substrate molecule. But according to induced fit theory, Koshland says that your enzyme is not rigid, but it is flexible. Enzyme will be changing its structure according to the substrate molecule. So he said that enzyme is flexible, whereas lock and key model told that enzyme is very rigid. Let's see the figure. There is an enzyme molecule. Now, two substrate molecules are coming to bind with this enzyme. The enzyme has changed its orientation or changed its shape according to the substrate molecule that is coming to bind to it. Ultimately, the substrates are coming and binding to the enzyme to form enzyme-substrate complex. And thereafter, product is formed, which is released outside. And again, this enzyme is regaining back its original structure. Now, I want you people to just see this video once.
Let us see how enzymes increase the rates of chemical reaction. There are two models of enzyme action. They are lock and key model and induced fit model. According to the lock and key model, when the key or substrate fits the lock or active site, the chemical change begins. But the active site, the enzyme, doesn't change shape. However, modern X-ray, crystallographic and spectroscopic methods show that in induced fit model, where the enzyme changes shape when the substrate lands at the active site. This visual shows you the enzyme and different shaped substrates. Click on Next to see with which substrate the enzyme interacts. As you can see, the enzyme cannot interact with the substrates that are red, violet, green and blue in color because their structure does not correctly fit into the enzyme. But once the enzyme recognizes its specific substrate, The enzyme and the substrate fit together perfectly. Then, the reaction takes place with the enzyme and later the substrate transforms into products. In induced fit model, the substrate induces the active site, the enzyme, to adopt a perfect fit. For clear understanding of induced fit model, you can think of a hand in a glove. Consider the glove to be the active site, which does not attain its functional shape until the substrate, in this example, the hand, moves into place.
Now we are moving towards 4 steps of enzyme action. What are the 4 steps of enzyme action? First and foremost step is one or more than one substrate molecule comes and binding to the enzyme, and then the enzyme wraps on the substrate at a special area called as active site, and the combination is called as enzyme substrate complex. And what is the third step? This catalysis is when the substrate is changed. It will be broken down or combined with another molecule to make something new. If it will break or bend chemical bonds. When done, you will have the enzyme product complex. And coming to the 4th step, the enzyme releases the product. When the enzyme lets it go, it returns to the original shape. It is then ready to work on another molecule of substrate. So, until now, we have learned about what are the main differences between a chemical catalyst and an enzyme. I want you people to go through these links in order to learn more about the differences as well as the models like lock and key model and induced fit model. Please work on these questions so that you can assess how far you have understood today's class and know more about these topics. Thank you.